So I'm very happy to introduce our speakers tonight, or this afternoon, Regan Stephenson and Anabella Marie Galang. And I'm very excited to have them. And I'm not sure, who do I hand it off to first?
I'll take it away. Thanks.
Okay, sure, go ahead.
So I'm Regan Stephenson. I'm a fifth-year Ph.D. student at Cornell. My research is in cancer immunotherapy in the Fischbach Lab, the same as the presenters last month. And I'll let Anabella introduce herself too, and then I'll take it away with the past and present of immunotherapy.
Awesome. Thank you so much, Regan. Hi, my name is Anabella Galang. I am a sophomore at Cornell University, specifically the College of Agriculture and Life Sciences. I'm a biological sciences major concentrating in genetics genomics development, and I work in a cancer research lab focusing on testicular germ cell tumors here at Cornell.
Great. Okay. Okay, so I will take it away. So maybe you're unfamiliar with immunotherapy, and in order to explain the usefulness of immunotherapy, it's important to keep in mind the context of, keep immunotherapy in the context of other cancer therapies. So you might be familiar with some of the other ways cancer is treated: surgery, radiation, and chemotherapy. These treatments all work to remove, kill, and slow down cancer cells, but depending on a patient's case, sometimes not all of these are options, and maybe some will be used together without one or the other. So for example, some tumors are inoperable, while others are resistant to chemotherapy, like for instance in some brain cancers where chemotherapy cannot pass through the blood-brain barrier. So as a result, immunotherapies have been introduced within the last few decades to fill the gaps that some of these other treatment options have. And some work to help support these other options, surgery, radiation, and chemotherapy, while others worked as standalone treatments to kill cancer on their own. It's important to keep in mind that immunotherapies may not be a good option for certain patients, and they're currently only approved for a few very specific, handful of cancers at very specific stages. But it's a really exciting new field of research and a new way to think about getting rid of cancer. So in this presentation, we're going to outline the past, present, and future of immunotherapy. So in order to understand where the field is headed, it's important to think about where we've come from and how it's based off of the immune system. So in order to understand immunotherapy, we must kind of understand immunology to start off. So when we think about getting infected with a pathogen such as coronavirus or a bacterial infection, we have two arms of immunity that come in to resolve our infections. The first is innate immunity, which starts early by itself, and then but has a broad application that's not very specific, and so it's a good band-aid on the problem, but adaptive immunity comes in a few weeks later, that's more specific and pathogens targeted, and so that eventually the combination of the two is actually what gets rid of our infections. So just to summarize, innate immunity is fast, consistent, but has a limited toolkit for recognizing pathogens. And this can also be applied when we think about how the immune system gets rid of cancer. So adaptive immunity, while it's slow and variable, it's teachable, so that it's something that we can mold and engineer to specifically get rid of certain pathogens and cancers. And that's exactly how immunotherapy works. So the immunotherapies that we have now use both the innate and adaptive immune systems. And these are some of the main players in immunotherapies currently. Maybe you've heard of antibodies in the media these days because they're important for getting rid of coronavirus, we think. And then some immune cells, macrophages, natural killer cells, and T cells. These are all white blood cells that are found in the immune system. And so current immunotherapies use these different cells and antibodies to kind of update the immune system to make it faster and or cancer specific. And that's how we can use the immune system to fight cancer. So to give you, now that you know how the immune system roughly is used for cancer treatment, it's, let's talk about how this all got started. Here I've shown a timeline with very tiny font, so it's totally okay if you can't read it. That kind of shows just how early some of these ideas came about, you know, early 1800s. And these dark bluer bubbles are all FDA-approved therapies. So it's really only been in the last 2 or 3 decades that these have become well-known therapies for cancer, but these ideas started early on. So just to single out a few that can kind of express how all of this works, in 1866 and 1896, there was anecdotal descriptions of decreases in tumor size associated with severe infections of erysipelas and influenza. So patients were getting severe infections with other pathogens, and miraculously their tumors were going away. And so this kind of got researchers thinking, well, maybe the immune system just needs a jump start. So how can we use that idea to treat cancer? And Dr. William Coley used this idea a little bit later through the injection of bacterial foreign signals to jumpstart the immune system. So what he did is it sounds pretty crazy, but at the time, he injected bacteria that caused an erysipelas infection repeatedly into a man's throat tumor. And while this man had been told he had just weeks to live, with this treatment, Dr. Coley gave him 8 years, which was pretty miraculous at the time. And so he ambitiously went on and tried to inject many more people with tumors, and had very inconsistent results, and in some cases ended up killing his patients with the infection rather than the treatment. So that wasn't a great cancer treatment, but it did have some surprising results in a few patients. So later, instead of injecting live virus, he introduced bacterial toxins, was his final form. So just bacterial components were injected near tumors. And he did have some decent effects, but the FDA finally outlawed it in 1962 because it had some inconsistent results and some patients died. So while this may sound like a crazy idea, the basis of it is we need to wake up our immune system to cancer because it has all of the tools to eradicate tumors. It just needs kind of that reawakening moment. And so a similar method that's actually in use today in modern cancer therapy is BCG therapy for bladder cancer. This is Bacillus calmette-guerin. It's a French name. It's specifically the introduction of a non-pathogenic bacterial strain into the bladder to initiate an immune response during early stage treatment of bladder cancer. So how this works is bacteria is introduced directly into the bladder, and there's a zoomed in picture here. And what this does is it is recognized by the local immune cells in the bladder wall that are then, let's see, there we go, infiltrate towards where the tumors are, and then can cause remission of the tumors in bladder cancer that's in this epithelial layer. It's important to note that bladder cancer that happens in these lower layers is not treated with this therapy because we need to have close contacts between our bacteria and our tumors and then the immune cells. So this is a pretty interesting therapy and maybe you haven't heard of. And it works for bladder cancer because it's so accessible. We can have bacteria so close to our tumors. So while this won't work for a lot of different tumor types, we have some other tools for that. One of them being monoclonal antibody therapy. That's a mouthful, but basically it means identical copies of antibodies for the treatment of cancer. And to understand how it works, we have to go over the different parts of antibodies. There are these Y-shaped protein molecules that are found in the blood and on mucosal surfaces in the body, such as the airways and GI tract. And they're specifically used to tag pathogens, or in this case, cancer and tumor cells. So it has this Y-shaped, these two arms that are cancer specific, and then a consistent trunk at the bottom that can connect to immune cells. So you have a tumor specific end on the top and immune cell specific end on the bottom. And what this does is it brings the tumor cells near to the immune cells so that they can act on our tumor cells. And so these can work to kill cancer cells in 5 different ways, and that's what I'm going to go over next. The first way is by, like I said, bringing the immune cells close to the tumor cells. These are typically natural killer cells or macrophages. And so our tumor cells are tagged with this antibody to be eradicated by our immune cells. And once they're close together, our immune cells release enzymes which break down the tumor cells and in some cases actually eat the tumor cells themselves. So our first way is with immune cell help. Our second way of eradicating tumor cells with monoclonal antibodies is through immune proteins known as the complement cascade. So here I've pictured them as orange proteins. What happens is once our tumor cells are tagged with antibodies, they recruit in these orange proteins that cause holes to form in the outer membrane of our tumor cells. And what happens then is these holes basically all of the contents within the tumor cells get leaked out, and these tumor cells die because they are full of holes. So that's our second way. Third way is through mechanisms we don't quite understand. In some cases, direct binding of antibody to tumor cells causes direct killing. So that's an added benefit of monoclonal antibody therapy. Our fourth way is with ammunition. So engineers have been able to tag our antibodies directly with particles emitting radiation or even chemotherapeutic particles. And the idea behind this is that we are directly taking the drugs to the tumor cells themselves. So this way we have a lot of, or high concentration of our drugs near our tumor cells, where the healthy cells maybe near your tumor aren't getting affected by these drugs. They're having a lower concentration of drug nearby. So less off-target effects. And our last way is through starvation. Monoclonal antibodies can be used to block certain proteins that are known as receptors on our tumor cells surfaces. And so when they're blocked, hormones and nutrients can't pass through our receptors to cause added growth within our tumor cells. So that is, and without these hormones and nutrients, cancer cells can't grow as fast, and then sometimes they die off. A couple of examples of monoclonal antibody therapy that maybe you've heard of, one being Herceptin. This is an immunotherapy that blocks cancer cells that have a HER2 receptor from getting the hormones that they need to help them grow. HER2 is found on some breast cancers and ovarian cancers, and which makes them a great candidate for this type of therapy. It's important to note that HER2 isn't expressed in other cancers, so this would be a useless option in different kinds of cancer that don't have HER2. A more broad example of monoclonal antibodies therapy is Avastin, which works for all types of cancers because it's not dependent on HER2. What it does is it blocks the signaling used to form new blood vessels that provide veins and arteries for tumors to get the blood and oxygen and nutrients that they need. And so without the ability to build new blood vessels and feed our tumors, the tumors end up shrinking and in some cases dying off. And so this is an exciting option because all tumors need blood vessels, or most of them, I'll say. need blood vessels. And so that's a common trait that we can use to target a lot of different cancers, not just breast and ovarian cancer in the previous example. And this Avastin therapy has been FDA approved for treatment of glioblastoma, ovarian cancer, colorectal, and lung cancer so far, but has possibilities for other treatments as well. So I wouldn't be doing justice if I didn't mention checkpoint inhibition therapy as a very specific type of monoclonal antibody therapy that has received a lot of praise within the last 5 to 10 years. This had won the Nobel Prize in Physiology or Medicine in 2018, the prize was shared by these two researchers, Tatsuku Honjo and James Allison. They both discovered each discovered a different way that blocks or reawakens T cells to kill cancer. This is a complicated process, but I have a video next to show you how this PD-L1 therapy actually works. And I'll try and get that to play now.
As a scientist, I specialize in cancer immunotherapy. My work, and that of my colleagues, is to figure out ways to stimulate the immune system to kill cancer. To understand this concept, let's talk a little more about our immune system and how it works. Taking a look inside our body, all of us are born with an immune system that acts like the security checkpoint at the airport. Our immune system is like a high-tech full-body scanner system that protects us from things that are harmful. In the immune system, T cells have receptors that scan cells to distinguish between normal cells and abnormal cells, like virus-infected cells. Once detected, abnormal cells are attacked and removed by T cells. T cells also identify these cells for future recognition. The same T cells also play an important role in protecting us from cancer. Cancer cells are normal cells that have mutated. The more mutations cancer cells have, the more easily they are detected. Until recently, we didn't understand why T cells were detecting these cancer cells, but not destroying them. But on a closer look, we've made some revealing discoveries. We've learned that one of the ways that cancer cells get past the security system is with the help of a molecule called PD-L1 that is found on the surface of cancer cells. PD-L1 is almost like a disguise that allows cancer cells to remain undetected. PD-L1 basically hacks the T cell scanning system by jamming the signal to parts of the T cell system called PD-1 and B7-1. Think of PD-1 as a sensor that's part of the T cell scanning system, and B7-1 is like an alarm that issues an alert when something is wrong. When PD-L1 hacks the system, it prevents it from doing what it's supposed to do. In other words, it may prevent T cells from destroying cancer cells. Scientists have been working on fixing this problem and have designed molecules to target the PD-L1, PD-1 pathway. You can think of cancer immunotherapy as kind of like a software update for your immune system. Early studies suggest that by targeting the PD-L1, PD-1 pathway, T cells may be able to detect and destroy cancer cells. Let me show you in more detail. T cells have receptors that allow them to recognize cancer. Cancer cells can prevent the elimination by turning on PD-L1. PD-L1 deactivates T cells by binding to PD-1 and B7-1. Blocking PD-L1 from binding to PD-1 and B7-1 could prevent PD-L1 from communicating with them. When this happens, T cells may finally be able to respond and kill cancer cells. Clinical trials are currently underway to understand the effects of trying to block the PD-L1, PD-1 pathway in the body. We are committed to studying cancer immunotherapy so that we can continue to find new ways to harness the immune system to fight multiple types of cancer. Brought to you by Genentech, a member of the Roche Group.
Okay, so now that we've covered checkpoint blockade therapies, let's move on to cancer vaccines, another way that the immune system is used for cancer therapies. Okay, so the first way that vaccines can be used against cancer is through the prevention of cancer in the first place. There are several viral and bacterial infections that can lay the foundation for cancer to arise later on in life. Some of these you may be familiar with on this slide: hepatitis B and C for liver cancer, tuberculosis for lung cancer, human papillomavirus for cervical cancer, as well as a variety of others. And because we know that these pathogens can cause cancer later in life, vaccines are used to prevent the infection from occurring in the first place, so we can prevent cancer from ever happening. Some of these vaccines you may have heard of, such as Hep Sal V against hepatitis B, which would hypothetically prevent liver cancer, as well as Cervarix and Gardasil for HPV-related cancers, such as cervical cancer, anal, head and neck, penile, throat, vulvar, and vaginal cancers. So these vaccines all work to prevent cancer, like I said, from ever happening. But what if we could use vaccines to eliminate cancer like it's a pathogen? And that's where the newest cancer vaccines are coming in. This is a cellular vaccine known as Provenge that's used for castration-resistant prostate cancer. And how this works is a patient's blood is taken, and from that blood we get cells called monocytes. Then in the lab, genetic material is introduced into our monocytes, which then create this Provenge cellular vaccine. And so these cells now know that prostate cancer cells are bad news and that they should be eradicated. And so once we have these engineered cells, we can put them back in the patient's body that pass on this news that cancer cells are bad to our resting T cells in the patient's body that are just waiting for news like this to come in. And then once the news is handed off, these T cells, these immune cells in the body are now cancer-seeking activated T cells that can then go on to eliminate cancer by themselves. So this is a long and complicated and expensive process, but ideally or hypothetically, if we just change out this genetic material, this process could be used to fight a variety of different cancers. Right now, it's only an option for prostate cancer, but it could be a pipeline used in the future. And with that, I'm going to let Anabella take it over to tell you all about the future of cancer immunotherapy.
Thanks so much, Regan. Okie dokie. We're going to go ahead and get started. Thank you for that beautiful first half. So we're going to be talking about the future of immunotherapy. Oh, first of all, can I get a quick nod if you can see the slides? Stellar. Thank you. Okay, future of immunotherapy. Let's go. So we're going to cover 4 different aspects today, starting with cancer vaccines, which Regan lovely left us off on. So vaccines have had a variety of applications in the past. In fact, it's nearly eradicated smallpox, polio, measles, a number of extremely difficult diseases that humanity has now largely overcome. And how did it do that? It increased a patient's immune response. It did this by not only informing it of what it needs to be looking for, but also strengthening the overall response. So how can we apply this to cancer? Well, that's a great question. So it involves, first of all, understanding the way that our bodies recognize the cells within. And that's accomplished through these things that are on the outside of cells called antigens. So one way to increase the level of recognizing said antigens would be to simply increase the number of antigens present in the system. Now, how could we possibly do this? Well, we could either do a direct antigen injection where we just add more of said antigens, or we're able to lyse agglomerations of cancer cells in order to expose more antigens, because the more exposed to those said antigens, the more the more accustomed to seeing the immune system will be. But here's the big question. So cancer cells arise from our own human healthy cells. So how do we know which are the right antigens? Well, those would have to be antigens that are specific to solely the tumor at hand. And those antigens are known as neoantigens, and neo means new. And so those are the antigens that are new when it comes to the formation of the tumor. Now, how are we supposed to figure out what the neoantigens are? First of all, we have to sequence the individual's tumor. So one of the interesting things about cancer vaccines is that they have to be specific to each individual. Even if people have the same type of cancer, the makeup of the cancer, the blueprint that guides it, is different for each individual person. And so if we wanted to be able to target an individual specific tumor, we would have to sequence it for each patient. So after we sequence it, we would be able to identify the neoantigens within the overarching blueprint, which is the DNA. So once that DNA is had, we can turn it into merely the mRNA. So the mRNA is kind of a spin-off of the DNA that's specific to help make proteins. So once that mRNA is synthesized, dendritic cells can take it up and become hardened. They become smarter. They know what they're looking for because they now understand what proteins are specific to these cancer cells that are causing so many difficulties. So now that we've got these more intelligent dendritic cells, they're able to create that increase in the patient's immune response that we've been looking for this whole time. So moving from cancer vaccines to oncolytic viruses, and what a time to talk about viruses. So first, to start off, we're going to talk a little bit about SARS-CoV-2, which is the novel coronavirus that is currently plaguing our world today. We are going to use this comparison to learn a little bit about the ways that viruses work, and then we'll translate it over to talk about oncolytic viruses. So SARS-CoV-2. It is a novel coronavirus, like I mentioned, that targets specifically cells in the lungs. And there's a little crown on it to demonstrate that it's called a coronavirus because of these spike proteins present across the surface of it. Now, that's the coronavirus's own version of antigens because that's the way that it is well received by the lung cells. So those spike proteins, specifically for SARS-CoV-2, hops onto something called the angiotensin converting enzyme 2, also known as ACE2. And this is present on a lot of your lung cells, and it plays a really big role in your ROS system, which is used for regulating your blood pressure and a lot of essential functions within your bodily systems. And so what your lung cells do is they engulf the coronavirus because, well, it believes that it's supposed to once it attaches onto that receptor. And once the virus is inside said lung cell, it's able to infect it and hijack its mechanisms in order to make proteins that will do its bidding and spread to other cells. Now onto a virus that actually might be beneficial to have in our system. So oncolytic viruses, first of all, what even are oncolytic viruses? So first, let's break it down. So onco is a prefix that means like abnormal growth, or in our intents and purposes, it means a tumor. Now, lytic refers to lysis, or kind of the destruction or like explosion of a certain entity. So we've got cancer and we've got like a lysis sort of situation. So what does that mean? We're going to investigate a specific example. So a recombinant poliovirus has been used to treat recurrent glioblastoma, which is really just a fancy word for brain cancer. As a matter of fact, it's the most deadly and common brain cancer out there. So what scientists were able to do were edit a poliovirus in order to go in a non-lethal way, target these glioblastoma cells, and overall be able to decrease the mass of the glioblastoma in patients' tumors. So on a survival curve, if we look at this, so patients who had the recombinant glioblastoma, they survived at a 20% rate, whereas the control fared much poorer. So how does this look on an overall like visual level? So first of all, this is a baseline MRI, and you can see it, the glioblastoma right here. And this is 2 months after the poliovirus infusion. And this is 6 months after, 12, 24, and 36 months following. And so what we've seen is an overall decrease of the mass of the glioblastoma. And this is particularly interesting because in the past, immunotherapies to treat brain cancers was originally unheard of. Why? Because there's something called the blood-brain barrier, which is it's an evolutionary like device within humans where in order to protect the brain, because it plays such an essential role in our life, it actually knocks down the immune system's abilities within your brain. So using an immune-based therapy in a space that's really highly policed for your immune system doesn't exactly play out as well. But in this instance, they were able to find a treatment that actually worked. And so this poliovirus has the capability to help decrease how dangerous glioblastoma are. So next, we are going to touch on solid tumors. So this is a picture of some histology for a solid tumor. I believe this is an ovarian, this is a mouse ovary actually. And so this is just to bring up a specific point. When it comes to solid tumors, immunotherapy sees a little bit of a challenge, and here's why. If you look at these cells, they are super tightly packed. And as formally explained, there's so much of immunotherapy that's reliant on surface area. The more bulky something is, the less the ratio between surface area and volume. And since the immune system needs to be able to recognize it, well, we need to have more surface area. So in the instance of solid tumors, it presents a particularly interesting challenge for the field of immunotherapy to cover in the future. So going from solid tumors to CAR T-cell therapies. First of all, what are CAR T-cells? Thank you for asking. So the CAR in CAR T stands for chimeric antigen receptor, but what does that really mean? So first, let's touch on chimeric. So the chimera it originated in Greek mythology, and this statue is actually a beautiful, teeny tiny bronze statue. It's currently in Florence, and this Etruscan piece of art depicts it's a she-wolf who also has a number of different animals like depicted here. And this speaks to the modern day translation of chimeric, which is a mixture of genetically different tissues. So a more tangible example would be if somebody had a kidney transplant. That would technically be chimeric, like a chimeric tissue. So antigens, as we talked about earlier, are these guys who reside on the cell and help people recognize, well, not people, help entities recognize it. And receptors are the counterpart to antigens. So they're the things that look for and detect specific antigens. So how are CAR T-cell therapies made? So similar to cancer vaccines, CAR T-cells are also specific to patients. With that in mind, we need to first collect T cells. So we talked about the CAR, now it's time to talk about the T. So when it comes to T cells, they need to be collected by apheresis, which is just a fancy term for drawing blood. Now, a few caveats with this is that many cancer patients or people undergoing any sort of cancer treatment are immunocompromised, particularly in instances with leukemia. They actually might have to pause treatment in order to collect sufficient blood in order to create a specialized CAR T-cell therapy. So next, we're going to touch back on the CAR. And that again is just the chimeric antigen receptor, which scientists create in a lab and then infuse onto the T cells that they collected from the patient. And all in all, you add them all up, you have the beautiful and fierce CAR T cell, which has immense capabilities and is currently being used in a number of different types of lymphoma in clinical trials. So what are a few considerations for the future when it comes to this really exciting new innovation? Okay, so the first European CAR T cell meeting actually just happened last spring, right before, one of the main interviews occurred with a woman named Regie Verhoeven, and she talked about a number of different concerns and ways to look when it comes to CAR T cells. So first and foremost, predictors for who is the best patient remains unknown. So what does this mean? That means that we're not quite sure at this point in time who's going to respond best to this type of treatment. And we need to figure that out so that we're not drawing blood or doing any sort of experimentation in vain. Next, long-term safety is not characterized very well. And so this is another important point to bring up. The truth is that CAR T cells have been around on this earth for a shorter time than I have. They were first made at Memorial Sloan Kettering in 2002, and they have not been around for a very long time. And as a result of that, we don't know its long-term effects because there have been no longitudinal studies beyond 15 years. And lastly, it's extremely important to listen to patients with how they experience their treatments. Because it's such a new innovation, it's pertinent that we undergo these clinical trials and really listen to everyone who tries this new therapy so that we can best revolutionize it for generations to come. And so those are my 4 sections, and that was the past, present, and future of immunotherapy. And at this point, I would like to open the floor up to any questions people may have. Thank you.
Thank you, that was terrific. I have one question to start off with. I know that sometimes I hear that melanomas respond well to immunotherapy because they're hot tumors, but I'm not quite sure what is a hot tumor.
Regan, I think,
Would you like me to take that one?
Sure, you can go ahead.
Yeah, so I think in Anabella's section, she explained that some tumors have neoantigens, and how that happens is in some tumors because of external stimuli, like for example in melanoma, from UV rays from the sun, we have a lot of mutations that cause melanoma to happen. And these mutations cause neoantigens. So they're protein sequences that aren't normally found in the human body. And so these hot tumors look different than, or look more different than healthy tissue compared to, less hot tumors. And so some of those tumors are easier to treat with immunotherapies because it's easier to click on the immune system because they're already so different.
Yes, and if I may follow up on that. So when it comes to hot tumors, that refers to tumors who have like a high like immune presence in that space. And so when it comes to skin, like skin itself plays a role in the immune system. And since it is like one of our primary barriers in fending off against pathogens, it makes sense that it would be relatively easy for those existent like immune cells to go in and take care of the melanoma. A contrast to that would be like a cold tumor, which for example would be like that glioblastoma I was talking about, in addition to like pancreatic cancer per se, where it's less likely that your body will be like, wait, whoa, do we really want to attack that? That seems kind of important. But if there's already immune cells there present, it makes more sense that it'd be willing to go for it.
Good, thank you. We have a couple of questions that came in on chat. Are you able to see those okay? Or should I read them?
I can take the first one from Sarah. Her question was about CAR T-cell therapies. With them being so new and with so many questions, why would insurance companies want to pay for that? The truth is they are very expensive therapies in the millions range, millions of dollars. And so I think to understand why they would be used, you have to understand when they would be used. And it's really as a last option for most of these patients, from what I understand. So we value human life, and sometimes you can't put a dollar sign on that. And so as a last therapy option, insurance companies will cover CAR T-cell therapies. I'm surprised that that happens, that they're accessible in Tompkins County, but that's really great news. From Matthew, what is the progression from academic research to industry in producing some of these products for the clinic? Is there a coordination between academic labs and companies like Genentech? Typically there is some collaboration. I'm not a part of any at the moment, but usually academic labs have a wide range of skills that they can answer some specific questions for companies for some of these products. So research labs at Cornell, for example, would be interested in the early stage research in some of these products. Later down the line, these giant companies like Genentech will do their own research, but sometimes specific labs have the right tools to do work with some of these big companies, but it's not as common as you might think.
Also, there was one point in time where I was interning in a in a more industrial setting, and one of the things that they always did was run their methods and everything by a university's lab before actually going through and doing their experiments. And I feel like that's not as common as it's not as common. But I have seen some more intimate collaborations between academia and industry.
I think it depends on, so yeah, Annabella's example would be a professor at Cornell being a consultant for some of these these giant companies where they can run your ideas by an academic. I guess from my experience as a student, I'm still learning. So some of these companies don't want to pay me to do their research, but maybe someone with a little more experience would get jobs like that to do some collaborations with these companies.
Can I ask what you both hope to do in the future?
Sure. So I'm working on my PhD. Who knows how long that's going to take, especially with the pandemic. But I'm hoping to work for a biotech company on immunotherapies. My current work is on macrophages in breast cancer. So I'm hoping to apply or work for some kind of checkpoint inhibition company, or maybe even CAR T at one point. So what about you, Annabella?
I'm currently pursuing my bachelor's degree, so I hope to get that. But afterwards, I am interested in pursuing an MD PhD program. I'm really interested in the physician scientist route, and I think it's really important to have both the breadth and the depth that provides. And I'm also really passionate about science communication, so that's something I would also be interested in, for the long term.
Mm-hmm. Very good. Any other questions right now? Well, if not, thank you both. That was really terrific. I mean, it's really hard to. I always find that immunology is very hard to explain and to do it very clearly. And those are both terrific presentations. So I really applaud you, and I appreciate you taking the time. And I know how much energy that takes to prepare these things. So thank you. Thank you very much.
It was my pleasure. Thank you.
Thank you.
Thank you.